Hi class. Today we're going to be talking about the origin of life. This is the second lesson in evolution. Sorry, I'm feeling a little ill today. What I want you to do first, and you can pause the video to do this, I want you to write down what you already know about the origin of life, what you want to find out about it, and at the end when we're done, you can write down what you learned. The essential questions that we're covering today are: what is the difference between spontaneous generation and biogenesis? What might have been the sequence of events that led to cellular life? And what is endosymbiont theory? First, let's look at our review vocabulary. We all know what an amino acid is. Those are the building blocks of proteins. Those are going to be essential to the creation or the origins of life. New vocabulary will be spontaneous generation, the theory of biogenesis, and endosymbiont theory. First, let's talk about the early ideas of where life came from. For quite some time, people thought that life arose from non-life, so spontaneously, life would come into existence. This was because you would leave a piece of meat out, you just saw the meat there, and then you would get maggots on the meat as it decomposed. They weren't seeing the flies coming and laying eggs on it and then the maggots coming from that. So a scientist, he runs an experiment. He puts a piece of meat in a flask and he leaves it open to the air. Over time, it gets maggots in it. He takes another flask and he closes it off, and over time, there's no maggots in it. So he shows that the meat does not spontaneously generate maggots. Some people didn't believe that. They said that they needed the air in it for the spontaneous generation to work. So Louis Pasteur designed another experiment. He takes this flask here, that has this long tube here, and you see this little curve at the bottom. The curve at the bottom is going to catch any microorganisms, but it's going to allow air to get in. So we're not going to have any microorganisms getting into it, only allowing air in. So he takes some beef broth and he sterilizes it, so make sure there's no organisms in it. And over time, he lets the air go through and no microorganisms are produced. Therefore, he proves that life comes from existing life. It does not spontaneously generate. So he was able to develop the theory of biogenesis. And that's what we accept today, that all cells come from pre-existing cells. All right, so that's once cells are here. Let's talk about the modern ideas of how we ended up with getting the initial cells. There are some that hypothesize about the primordial soup, where we have the Earth's atmosphere, and we have certain gases and things like that that help give rise to organic molecules. The sun and the amount of UV light and all of these helped build the very first molecules that were necessary to have life on Earth. Mueller and Urey, they did an experiment that actually showed that you could build organic molecules out of the environment that was early Earth. So they got in the lab. You see here we have the electrodes here. They're simulating lightning. We have your boiler here. So then you have the water that's going to go around. It's going to have the other gases that are prevalent in early Earth, and then they collect it down in the flask or the beaker down at the bottom, and it showed that there were organic molecules in there, specifically amino acids. So the first things that came about were amino acids. This is a video about their experiment. I've posted it on Schoology. You can watch it there. So we have these amino acids that are coming together. We know that amino acids in and of themselves are not going to do anything. We actually have to have them together to make a protein. So the idea is that there's small clay particles that allow the amino acids to get close enough to each other to allow them to assemble to each other so they don't break down and actually forms a protein. Otherwise, the amino acids, they come together, they hit each other, they bind, then they break apart. So the clay was allowing them to bind together and stay bound together. So after we've had the amino acids coming together, we have to find ways to code for things. So you know that we have the DNA that codes for things and the RNA that's carrying the message. Well, the DNA is way more complex than the RNA. So the idea was that the RNA was the first coding system and DNA came later. So that's leading us up to the first types of cells. So we have the RNA, and it's going to code for a membrane. So we're going to end up with a membrane-bound cell. So it makes a plasma membrane. Okay. So the first cells that they hypothesized were prokaryotes. They were very small, very simple in comparison to eukaryotic cells. They're going to be very closely related to archaea. So archaea, in its name, it's archaic. These are the really, really simplistic ones. They are going to be found in extreme environments, extremely hot, extremely acidic, extremely salinic. They've also been termed extremophiles. They were photosynthesizing about 1.8 billion years ago because there was no oxygen in the air. So they made their energy from the sun. There were cyanobacteria. The oxygen that they release from photosynthesis is the oxygen that we find in the ozone layer now. So oxygen-free Earth. We have these small organisms. They're going through the process of photosynthesis. They're releasing oxygen into the atmosphere that's going up into the atmosphere and creating the ozone layer. Ozone is just O3. All right, that leads us to endosymbiont theory. Endosymbiont theory says that there were eukaryotic cells that engulfed some early prokaryotic cells, and those early prokaryotic cells developed or evolved into organelles. So chloroplast and mitochondria, those are said to be early prokaryotic cells. This is backed up by them having their own DNA and their own existential membrane. So they're membrane bound and they have their own DNA. It was more beneficial to the prokaryotic cell to be inside of the eukaryotic cell, and then the eukaryotic cell benefited from having mitochondria because it got energy. It also benefited from having chloroplast because then it was able to photosynthesize. This is a video about how that came about. It's posted on Schoology as well. You can go there and look at that. All right, so we talked about the differences between spontaneous generation and biogenesis. We talked about the events that led to cellular life and endosymbiont theory. All right, go here, take this quiz. You need to log into your Gmail account. It's your student ID at students.fwisd.org. See you on Monday or Tuesday. Bye.